about whether it's favorable to do. So a spontaneous reaction
Can anyone hear anything? Because I can't hear anything.
No, I can't.
No.
Nope.
Can you hear us?
There's like no audio.
Ice Raven.
Yo, what you want? Wait, who's calling my name?
Ice Raven.
Rip.
Can you guys hear me now? Can you guys hear me?
We can hear you.
Okay.
Ice Raven.
So basically, was there a place where I left off at that you guys couldn't hear?
I think it was like that whole slide.
whole
Not gonna
thing.
lie. I'm not sure. I'm sorry.
Oh, so thermodynamics.
Yeah.
Yeah.
Okay, so
Beginning.
okay,
My
I'll
bad.
do it. Oh, sorry, I didn't
You're
realize my
all
mic was
good.
muted.
No, it's okay. It's
Okay,
okay.
so thermodynamics is there's some important things you need to know for thermodynamics for AP Bio. First, you need to know the law of conservation of mass, which means that chemical reactions do not lead to a change in mass in a closed system, which means a closed system is basically where energy cannot be exchanged or mass cannot be exchanged. You have the first law of thermodynamics, which means that energy cannot be created or destroyed. That's also that you need to know the first two laws of thermodynamics. And then the second law is basically energy transfer always increases entropy in the universe. So every reaction, energy is lost as heat, which is heat is basically a disordered energy. Then also you have an endergonic reaction or an endothermic reaction, and that basically absorbs energy, which means that your reactant to product, more energy will, there will be more energy in your products. You also have an exergonic reaction, which releases energy. That means that reactant to product, your products will have less energy than your reactants.
So would that mean stored energy? Stored energy? Would that mean stored energy for exergonic?
It really depends on the reaction, but if you're talking about cellular respiration, then yeah, the energy released could be used to make ATP, for example. So yeah, you can use exergonic reactions to allow harvest the energy released to make like stored energy or something. Then energy coupling is basically when you use an exergonic process that releases energy to drive an endergonic process or an endothermic reaction, because endothermic reactions are generally they're not going to go on their own. So what you need is the energy release from an exothermic reaction to drive an endergonic or endothermic reaction. And this is the Gibbs free energy equation. I don't really think it has really popped up the AP biotest, but it's still pretty good to know. So basically, spontaneous reactions occur without energy input, which means that other than the activation energy, which is needed for every reaction, you don't need to put external energy for the reaction to go. And you have the free energy, or delta G, and that's the energy used to do work. And so a favorable spontaneous reaction would have a negative delta G. H is your enthalpy or heat change. So basically a favorable reaction, like an exergonic or exothermic reaction, is going to release heat. And you have delta S, which is a change in entropy. So basically, always for spontaneous reactions, you want increase in entropy. So that means that your delta S should be positive. Your temperature, and T is just your temperature in Kelvin. Oh wait, And so yeah, if your delta G is positive, then your reaction is not spontaneous. And if your delta G is a less than 0, which is negative, it means it's spontaneous, which means the reaction will proceed by itself. And so there are some crucial things you need to know about enzymes, which is, first of all, yeah, just some pathways, like a metabolic pathway is just a sequence of chemical reactions. Then the reactant and ends with a product. And the enzymes are catalysts that they help speed up the reaction rate by lowering the activation energy, as you can see in this diagram. And then that the substrate is basically the reactant that binds to the active site of the enzyme. And so an active site is basically the region of the enzyme where the substrate binds. Wait, can you guys still hear me?
Yeah, we can.
Yeah, we can.
Okay, and then so yeah, the active site is just where your substrate binds to your enzyme. And so that's why shape is so crucial to function, because your active site is where your reactants will bind to your because it's where your reactants bind to your enzyme. And so its reactive site is specifically fitted for the reactants that bind to the enzyme. And so yeah, when enzymes bind to a substrate, there's something called induced fit. And so what that means is that once your reactant binds to your active site, the enzyme will change shape in order to bind more fittingly to the substrate, or basically the reactant. And so there are 4 ways enzymes can help lower the activation energy. First, active site can allow substrates to become in a proper orientation, allowing for the reaction to proceed. Also, allows for the catalysis and helps produce the product. Also, it provides a microenvironment that helps with the reaction. So maybe some reactions are more favored in an acidic, basic pH. Enzymes can help provide that. And also, enzymes can also directly participate in the chemical reaction. The thing is that when enzymes participate, because enzymes do not get used up in the reaction. So after the reaction finishes, it has to proceed back to its original form. The thing is that denatured enzymes cannot act as catalysts because their shape is disrupted, which means that whatever was supposed to catalyze the reaction cannot proceed because the enzyme has lost its function. And so enzymes have an optimal temperature and pH where the enzyme best works. And then the thing is, even though the basic human body has a pH of like 7.4, not all enzymes have that optimal pH, because it really depends on where the enzyme operates. So like pepsin, which is an enzyme in the stomach, it works best at a pH of 2. And if you look at the diagram, you can see that the optimal pH of pepsin is 2, while another enzyme, its an optimal pH might be 8. So it really just heavily depends on temperature and pH. And some enzymes also have non-protein helpers called cofactors, and this basically just helps catalyze the reaction. And so you don't need to know much or in depth about cofactors, but yeah, it's good to know that if a cofactor is organic, they're just called coenzymes. So vitamins are good examples of coenzymes that basically help enzyme with catalyzing reactions. Is there any questions?
So like coenzymes would help speed up the reaction?
Basically, yeah, they basically help enzymes catalyze the reaction.
Okay, I have a question.
Okay.
Mhm.
If enzymes go back to their previous state, why does it have a maximum substrate concentration? Like why a reaction has maximum substrate concentration?
Oh yeah, okay, so at a certain point, basically there's a really high substrate concentration. What happens is your reaction cannot proceed any faster, and that's because if you have a high substrate concentration, once reactant to product quickly forms by enzymes, another substrate immediately goes in. So there's like a maximum rate for certain enzymes, and that's just based on your amount of substrate concentration and also your amount of enzymes. At a certain point where you have a lot of substrate, the only way to increase a reaction is by increasing your amount of enzymes. I hope that answered your question.
So like that would happen like through the use of operons, right, in bacteria? Like increasing the amount of enzyme?
Or no, it's just like when you're catalyzing a specific reaction, it's just for any enzyme, there's just a specific substrate concentration where by adding more substrate, you can't increase the reaction rate, and that's just due to the fact that enzymes always, if their active site is occupied, it can't get another substrate. So
Yeah, no, I understand that part, but what I'm trying to say is like if there's like a high enough concentration of substrate, like it'll start reacting with an operon to produce more enzymes so that they can catalyze the reaction and produce.
Yeah, so if there's a lot of substrate, depending on like your operon, it'll probably make more enzymes in order to like catalyze the reactions with the substrate. To your question?
Yeah, thanks.
Okay. So basically, now for regulation of enzymes, there's basically enzyme inhibition, which prevents the action of specific enzymes. And so you have competitive inhibitors, which blocks substrates, and basically it competes for the active site, which means it basically binds at the active site. And so the thing is that your inhibition can be overcome by just increasing the amount of substrate. And you also have non-competitive inhibitors, which don't directly bind to the enzyme at the active site, but binds at a different site of the enzyme, or an allosteric site. Allosteric site is just basically another site that's on the enzyme that is not the active site. And non-competitive inhibitors, they usually what happens is they alter the active site so that substrates cannot bind to the active site of the enzyme. And so the thing is, the inhibition in non-competitive inhibition cannot be overcome. It's permanent compared to competitive inhibitors, which can be overcome by just increasing your substrate concentration. And so non-competitive inhibition is a type of allosteric regulation, and allosteric just means that it's based on a different site that's not the active site. I'm not sure if you know cooperativity, but it's just basically that if you have an enzyme with multiple subunits, a substrate binds to one of the active sites, it leads to a change in all of the subunits so that they become more active. And it's crucial. I don't think it's really needed for AP Bio, but it just helps to know. And so a crucial topic in metabolic pathways is the idea of feedback inhibition. And that just means that the final product switches off the metabolic pathway by inhibiting an enzyme. And so in this case, you can see that isoleucine inhibits the first enzyme, which is basically threonine deaminase. And basically it just, basically what it does is the product will inhibit the first enzyme. And so that prevents the reaction from going. And so it's a pretty common mode of metabolic control. That's it for feed. And that's basically it for enzymes, I think. Yeah, so we're going to go on to cellular respiration now. Does anyone have any questions for this, or we can go on to cellular respiration? Respirate. Okay. So for cellular respiration, you need to know that it's a catabolic pathway, which means that it breaks down complex molecules to simpler ones. And fermentation is just a catabolic pathway that breaks down sugar without the use of oxygen, and I'll explain that more in later slides. And aerobic respiration is just when you use oxygen as a reactant that's consumed along with the fuel. In this case, it's glucose. And so you need to know a little bit about redox and oxidation and reduction. And so redox reactions are just reactions in which you transfer electron one substance to another. I think it got cut off there, but yeah. Then oxidation is when a substance loses electrons, like for example, when glucose goes to carbon dioxide. And reduction is just the addition of electrons from a substance. In this case, oxygen to water, oxygen is reduced. And so a crucial thing about cellular respiration is this thing that energy is harvested in steps. And the thing is, if energy were harvested all at once, you couldn't get all the energy efficiently because a lot of it would be lost as heat. So basically, what happens eventually is that electrons that are stripped from glucose, they're transferred to NAD plus and FADH2, which can allow for and NAD plus is basically a molecule that allows for oxidation and reduction. It basically can move between the states really easily. And so what you just need to know basically right now that respiration uses a stepwise energy harvesting process, that it does not release its energy all at once. Stepwise energy harvesting process, electrons do not lose that much energy. And also the electrons, they get transferred to okay, there's a typo here. It should be saying NAD plus, not NADH, because NADH is the reduced state. The stages of cellular respiration. This is just like a summary right now. I'll go more in depth later. So glycolysis is the first step. And then the thing is, because it occurs in the cytosol, bacteria also use glycolysis, since bacteria or prokaryotes don't have a mitochondria, obviously. So they use glycolysis, which occurs in the cytosol. So glycolysis is used in both eukaryotes and prokaryotes, and it's the first stage of cellular respiration that evolved. So glycolysis occurs in the cytosol, and it begins the degradation process by breaking down glucose into 2 molecules of pyruvate. And you don't need to know much of what they are right now, except that pyruvate is a 3-carbon molecule. And pyruvate oxidation is the stage between the citric acid cycle and glycolysis. And basically what that is that pyruvate oxidation occurs in the mitochondrial matrix, and it's where pyruvate is broken down into acetyl-CoA. And so acetyl-CoA, so basically the citric acid cycle is also in the mitochondrial matrix, and it uses acetyl-CoA, and what it does is it completely breaks down glucose to carbon dioxide. Phosphorylation is the last step of cellular respiration, and it occurs in the mitochondrial inner membrane. And ATP synthesis is powered by the redox reactions of the electron transport chain, which we'll go into later. It includes chemiosmosis and the electron transport chain. So oxidative phosphorylation is an umbrella term for chemiosmosis and electron transport chain. So this is glycolysis. So one molecule of glucose becomes 2 molecules of pyruvate, which are 3-carbon molecules. And 4 ATPs are made, but used in the steps of glycolysis. So basically the net production is 2 ATPs. And the thing is, for glycolysis, for any of cellular respiration, you don't need to know the specific cycles or the specific intermediates that occur, but you need to know what is produced. So you produce 2 ATPs. And so the thing is, the ATPs are made by substrate level phosphorylation, which means an enzyme transfers the phosphate group from a substrate molecule to ADP. And that's going to contrast oxidative phosphorylation, which we're going to talk about later. And it produces 2 NADHs. And no CO2 is released at this stage. Glycolysis occurs whether or not O2 is present. So it doesn't require O2, and so it always goes on. And this is pyruvate oxidation and the citric acid cycle. So pyruvate oxidation is when pyruvate, so first of all, pyruvate first enters the mitochondria by active transport. So a protein will transport pyruvate into the mitochondria. Then you have pyruvate that is converted to acetyl-CoA, which is a 2-carbon compound. And since pyruvate was a 3-carbon compound, that means CO2 was somehow released, and 1 carbon was released, and it's released in the form of CO2. And also NADH is produced per pyruvate, which means that you have a net of 2 CO2 and 2 NADHs from 1 glucose molecule. And because 1 glucose molecule makes 2 pyruvates, so each makes 1 CO2 and 1 NADH, so the net is 2 CO2 and 2 NADHs. So now you already have like a partial breakdown of glucose, but now going into the citric acid cycle, it's going to further break down glucose to carbon dioxide.
Wait, so there's no FADH from pyruvate oxidation, right?
No, it's just NADH.
Thank you.
And now you have the citric acid cycle. And then the thing is, you should know that's also called the Krebs cycle or the tricarboxylic acid cycle, because these are all synonymous with each other. So basically, the citric acid cycle makes 1 ATP, which is by substrate level phosphorylation using enzymes by transferring phosphate to ADP. So it makes ATP. It makes 1 FADH2 and 3 NADHs. And this is per acetyl-CoA. And since you have 2 acetyl-CoA coming in per molecule of glucose, in total, the citric acid cycle makes 2 ATPs, 2 FADH2s, and then 6 NADHs. And so this is a diagram I found for it. Basically, you don't need to know any of the intermediates like citrate or oxaloacetate. You should just know like NADH is 3 NADHs are made, 2 CO2s are released, and 1 FADH2 is produced, and then 1 ATP, or in this case GTP, which is basically the same thing, is produced. And so this is one turn of the citric acid cycle, which means since there's going to be, due to 1 glucose making 2 acetyl-CoA, so it's all of these products are going to be doubled. Does that make sense? Yes. Okay. Okay, and next we're going to go into oxidative phosphorylation, and that's basically the last step of cellular respiration. And so this is the pathway of electron transport. You don't need to know the pathway of the electron transport chain. That's like way too detailed. But you should just know what cytochromes are. These are just electron carriers in the electron transport chain. And it just contains this iron-containing group called a heme group, and that's an example of a cofactor, if you remember from enzymes. And basically what this does is it helps donate and accept electrons. And eventually the electrons are passed to oxygen, which is the most electronegative molecule, meaning that it accepts electrons. It has the strongest electron affinity. And so what happens is after the electrons are passed to oxygen, it forms water by taking 2 hydrogen ions from the surrounding. Each cytochrome has different electronegativity, and if you look at the electron transport chain, you'll see as it goes down the chain, the molecules, cytochromes, become more and more electronegative. And so next so as you remember, oxidative phosphorylation contains both electron transport chain and chemiosmosis. So chemiosmosis is the mechanism that it can harvest energy to produce ATP. So penetrating the inner membrane of the mitochondria, you have a protein called ATP synthase, which can help generate ATP using an H+ ion gradient. And the H plus ion gradient is basically formed from the release of energy from the electron transport chain, as stated here. It uses the exergonic flow of electrons, which is donated from NADH and FADH2. So electrons from NADH and FADH2, they donate their electrons to the electron transport chain, which it helps so the electrons basically release energy, and it helps to pump H plus across the membrane space. And it's pumped from the matrix to the intermembrane space, and that's an interaction you should definitely know that where the electrons are pumped towards, because I have a question. Oh, okay, so yeah, so basically use this H plus gradient to drive ATP synthesis, and that's called chemiosmosis. And so yeah, the electrons from NADH and FADH2, they release electrons to electron transport chain, which helps to pump H plus across the membrane space. And it's from the matrix to the intermembrane space. You should know how the H plus ions are transported. So the H plus gradient is called a proton motive force. And the reason it's called that is because a gradient can be used to drive ATP synthesis. And so yeah, not all active transport mechanisms also use ATP. They can also, in this case, the ion gradient can be used by the ion gradient can be made by, in this case, the releasing of energy from electrons. And so chemiosmosis couples energy in the form of an H plus gradient to drive cellular work. In this case, it's ATP synthesis. And so this is a really nice diagram on how everything works in oxidative phosphorylation. So you have this electron transport chain, which, as you can see, oxygen is the last carrier, and so it takes two H plus ions from the surroundings to make water. So H plus is transported from the matrix to the intermembrane space. And it's really important because I think on the 2019 multiple choice, I don't know, I remember this question where it was talking something about like which part of the mitochondria is the most acidic. And so in this case, it will be the intermembrane space because that's where hydrogen ions are transported to. And so yeah, after the H plus ions are transported to the intermembrane space, it'll go through ATP synthase, and as it diffuses down, ADP plus phosphate will become ATP. And yeah, and so also FADH2 and NADH donate their electrons to the electron transport chain. And so want to know how many ATPs are produced by cellular respiration. So each NADH molecule makes about 2.5 ATPs, and then FADH2 makes about 1.5 ATPs. So the ATP yield also varies heavily depending on a shuttle used to transport electrons from the cytosol to the mitochondrion. And what that means is, remember in glycolysis, you do make two NADHs. And the thing is, the NADHs from the cytosol cannot just go to the mitochondria. You have to transport it because basically you have two NADHs stuck in the cytosol, and it can't be made into ATP unless transported into the mitochondria. What happens is you have a shuttle used to transport basically the NADHs from glycolysis to the mitochondria. But basically the shuttle is just another NAD plus or FAD molecule in the mitochondrial matrix. This is important because FADH2 makes less ATP than NADH. So what happens is it's purely by chance whether or not shuttle uses NAD plus or FAD, sorry, in the mitochondrial matrix. And so basically, if the two NADHs are made from glycolysis, if they're transferred to FADH2, then they're going to make 3 ATPs. But if the NADH electrons are transported to NADH, then it will yield 5 ATPs. So in total, cellular respiration makes 12 NADHs, 2 FADH2s, and then 4 ATPs, which is by substrate level phosphorylation. And so if you calculate everything, the 12 NADHs, 2 FADH2s, and 4 ATPs, that in total will make 30 or 32 ATPs, depending on whether the shuttle is NAD plus or FAD. And does that make sense?
Yes.
Okay. So yeah, that's basically it for cellular respiration. We're gonna go on to fermentation, which is when you don't have oxygen. When you don't have oxygen, oxidative phosphorylation does not occur because instead glycolysis will still occur. But then so basically fermentation is a subset of another pathway to help produce ATPs when there's no oxygen. And so they also use anaerobic respiration, which is not the same. It's not. So the thing is, anaerobic respiration can be considered fermentation, or there's another pathway that prokaryotes use that doesn't use oxygen that is not fermentation. So there's like two definitions for anaerobic respiration. And in this case, it just anaerobic respiration occurs in prokaryotic organisms, and the equation has sulfur in it. So there's two types of anaerobic respiration. One is considered fermentation. Another is respiration that uses sulfur and does not use oxygen. And fermentation is glycolysis plus there's a reaction used to regenerate NAD+. And the crucial thing about fermentation is that it has to regenerate NAD+, because if you have too much NADH, the cell, it does not allow for cellular respiration or fermentation to occur. So you have to always regenerate NAD+, because you can't have too much NADH in the cell or it will stop cellular respiration. And the thing about fermentation is it really doesn't form that much ATP. It only forms 2 ATP. And then you have to add so it only forms 2 ATP. And so it basically doesn't, so it's just not as efficient as cellular respiration or aerobic respiration using oxygen. And so there's two types of fermentation. You have alcohol fermentation and lactic acid fermentation. So alcohol fermentation occurs in yeast and a lot of bacteria. And what it does is it produces 2 ATP. So first of all, when you have glycolysis, it produces 2 ATP, 2 NADHs, and 2 pyruvate. And so what happens is the pyruvate basically accepts the 2 H plus ions from NADH, which helps regenerate the NAD plus to form alcohol, so ethanol. So basically the pyruvate becomes alcohol. And also CO2 is released, and that's why bread rises, because you have yeast doing alcohol fermentation. And yeah, so the crucial thing is the pyruvate becomes alcohol and the NAD plus is restored when NADH transfers the hydrogen ions to ethanol. And also you also have lactic acid fermentation, which is when pyruvate also transfers, when NADH transfers to H+, except it forms lactate instead. And so it does not release CO2 and is lactate instead of ethanol. And ethanol is in alcohol fermentation. So yeah, and then so basically humans use lactic acid fermentation, and it's also why our muscles feel sore during exercise, because of the lactate that's produced. People should know obligate anaerobes, which means they cannot survive in the presence of oxygen, and they carry out fermentation or anaerobic respiration. And facultative oxygen, and they can make ATP through aerobic respiration or fermentation or anaerobic respiration. And so yeah, as it's stated here, glycolysis evolved early to make ATP. And so that's why it's the most widespread pathway among organisms, because all organisms need energy. And it's simple in that it can be used in the cytosol. It does not need a mitochondria for the processes to occur. And this is basically a good diagram for alcohol fermentation and lactic acid fermentation. 2CO2 are released during alcohol fermentation, and it becomes ethanol. And in this case, pyruvate becomes lactate. And you also need to know, like, basically the similarities between these pathways, that the NAD plus is regenerating both pathways. And that's, and then also for cellular respiration, you kind of want to know that also fats and proteins can be used in cellular respiration, and fats are converted to acetyl coenzyme A, and then it goes through the whole entire cellular respiration pathway. Proteins can basically be converted into most intermediates of the cellular respiration pathway. And so, yeah, you need to know, like, glucose isn't necessarily the only product that is made. Glucose isn't necessarily the only molecule that can be used in cellular respiration, that also fats and proteins can be used in cellular respiration as well. And so you should maybe know a little bit about the regulation of cellular respiration. So there's a protein called phosphofructokinase, which is in glycolysis. And what it does is it helps regulate the rate of cellular respiration. That's all you need to know. You don't need to know specifics about this, but you should know that there is some sort of regulation of cellular respiration. And that's basically it for cellular respiration. If you guys have any questions right now, ask me. Do you guys have any questions about cellular respiration? Or do you want us to move on to photosynthesis? Okay, so we'll move on to photosynthesis. Basically, photosynthesis is an anabolic pathway, which contrasts with cellular respiration, which is a catabolic pathway. And so anabolic pathway is a pathway that uses energy and it builds complex molecules, like glucose. And so chloroplasts are the site of photosynthesis, and so basically in plants, and they're basically abundant in the mesophyll, which is just the interior layer of plant leaves. You should maybe know a little bit just about the stomata, just because gas exchange occurs in here, which means that CO2 goes into the stomata, which you will need for photosynthesis, and O2 goes out, which is a byproduct of photosynthesis. You don't need to know specifics since plants aren't on the test, but it's helpful to know how they get all of these reactants and how products are excreted. And so so you need to know some basic anatomy about the chloroplast. So the stroma is the fluid in the chloroplast, as shown in the diagram. And thylakoids are basically these small, like, sacs in the chloroplast. And a stack of thylakoids are called a granum. And chlorophyll is just the light absorbing pigment is found in the thylakoid membrane. So yeah, and so basically in this case, CO2 gets reduced to become glucose, and water gets oxidized to form oxygen. So this is also a redox reaction like cellular respiration. And so you also need to know some basics about the electromagnetic spectrum, which basically states that visible light is 300. You don't need to know you don't really need to know, like, the specifics of the electromagnetic spectrum, but maybe you just know that visible light is within a certain range, like 380 nanometers to 750 nanometers, easily detected by the eye. Also, red has the longest wavelength, which means it has the least energy, and purple has the most energy, which means it has the shortest wavelength. And photons are basically just light particles. And so, yeah. And so you have an absorption spectrum, which basically graphs absorption versus the wavelength. And so basically, yeah, you need to know the main pigment in photosynthesis is chlorophyll A, and it absorbs mainly violet, blue, and red light. And so you also have accessory pigments, which are chlorophyll B and carotenoids. And not only do they help broaden the spectrum of which you can absorb light, but it also helps protect the plants against excessive light. So it basically helps protect chlorophyll from excess damage from absorbing the excess light. And so if you want to know, carotenoids are basically like orange-ish. And so it's pretty, but mainly it's really important that you know the pigments of photosynthesis. And also the accessory. Do you have a question?
Wait, so that's a good explanation for why plants aren't black, right?
Yeah, basically, like, or it's just because. What do you mean? That?
Like, I mean, like if plants are black, they could just accept all the light, and then they would have everything. But so instead, the chlorophyll B and carotenoids are to kind of make sure that they don't reflect too much and I guess burn out.
Yeah, because if plants were black, like, would suffer a lot of damage to absorbing so much light from the sun and everything. So it helps protect the plants from excess light. Because light is basically energy, so it could damage the plants.
Yeah, I know. I just thought that was an interesting idea that came into my mind. Thank you.
Yeah, no problem. And so the thing is, plants are green because the light is reflected. So yeah, that's why leaves appear green, because as you can see, based on the absorption spectrum, all of the absorbs no green light because all of the green light is reflected. So green light is not used in photosynthesis. And so you also have an action spectrum. And so it basically profiles the relative effectiveness of relative and different wavelengths. The thing is, your action spectrum looks different from your absorption spectrum because of the presence of different pigments, as I just said, like carotenoids and chlorophyll B. So that's why also the action spectrum is a lot more effective than the absorption spectrum, because it has different pigments that allow for different various wavelengths to be absorbed. And so it combines, basically, as I stated, it combines the effectiveness of different pigments, chlorophyll A, accessory pigments like chlorophyll B, and carotenoids. I didn't put a lot of information, but there's this thing called Engelmann's experiment. If you want to check it out, it might be really important. But just like a general summary, it's using photosynthetic algae and aerobic bacteria. And basically what he did is, in a time of like no technology, I think it was done like in the 1800s, he was able to figure out the action spectrum for photosynthesis. And I didn't include enough information here, but if you want, you can probably search it up on Google. Okay, yeah, but now we can go through the light reactions for photosynthesis. And so the light reactions occur in the thylakoid membrane, and it helps convert your light energy into chemical energy that will later be used in the Calvin cycle. So there's two steps of photosynthesis. You have the light reactions and you have the Calvin cycle. It helps convert light energy into chemical energy, like ATP and ADPH. And it splits, and it also splits water to help regenerate electrons. And also, this is the step where O2 is released. And so that's basically the general overview. So first of all, in a thylakoid membrane, you have photosystems, and you have photosystem 2 and photosystem 1. So photosystem 2, the difference between the two photosystems is photosystem 2 absorbs an optimal wavelength of 680 nanometers, and the photosystem 1 absorbs a wavelength of 700 nanometers. And so this is something weird, but like photosystem 2 goes first, the light reactions. I don't really know why it's called photosystem 2 because it goes first, but yeah, so.
I think it was like photosystem 1 was like founded first, or like photosystem 2. I don't know. It was like weird.
Photosystem 2 was discovered first, I think. No, wait, photosystem 1 was discovered first, and then 2 was discovered. Yeah, my bad.
But it's second in the process.
Yep.
Yeah. So basically, you have photosystem 2 that goes first, and then photosystem 1 goes second. So what happens is you have a photosystem that that consists of this reaction center complex, and you have light harvesting complexes. And so light harvesting complexes are they're basically proteins, and they contain, and that's the complex that contains chlorophyll. So basically sunlight is absorbed from the light harvesting complexes, which makes sense by the name. And so a photon from sunlight will hit a pigment molecule in the light harvesting complex. In this case, it's the pigments are just basically the chlorophyll A, carotenoids, chlorophyll B. And so it hits photosystem 2. And so basically the energy will cause an electron to be excited, bumped to a higher energy level. And it gets transferred to the primary electron acceptor, which basically the primary electron acceptor will later go to the electron transport chain. And the thing is, you have 2 electron transport chains, but yeah, we'll go into that later. And so the thing is, one of the electrons, basically one of the electrons are used because they're going to go to electron transport chain. So you have a missing electron. And so what happens is water becomes split in order to replace the missing electron. And so your excited electron, it passes through the electron transport chain, and kind of like cellular respiration, it pumps hydrogen ions into the thylakoid space, which later will be used to create a proton gradient again, like cellular respiration, and it helps in ATP production due to ATP synthase. So it really helps to know cellular respiration than photosynthesis because a lot of the processes overlap. And then the electrons, after going through the electron transport chain for photosystem 2, the electron will be transferred to photosystem 1. Then it gets boosted to a higher energy level, so it gets excited again by light absorbed from the sun, from the light harvesting complex. And so after it passes through another electron transport chain. And so this is photosystem 1. And so it will pass NADP plus will become NADPH. And so yeah, you need to know like that it's NADPH and not NADH, because sometimes I think people accidentally write NADH. And so yeah, basically the ATP and NADPH created from the light reaction will be used in the Calvin cycle. And so basically ATP and NADPH are basically stored energy that will be used to reduce carbon dioxide into glucose. And so yeah, this is just a simplified diagram of what I basically explained. Water hits chlorophyll molecules, which leads to the excitation of electrons, and it goes to a primary electron acceptor, in which it goes through electron transport chain, which makes ATP by proton motive force and ATP synthesis, chemiosmosis basically. And then it goes to the photosystem 1, in which basically light excites the electron again, and then the electron in this case is donated to the primary electron acceptor, where it goes to NADP plus reductase, which basically converts NADP plus to NADPH. And yeah, water is crucially split to provide electrons in photosystem 2, and O2 is produced as a byproduct. And yeah, this is basically another diagram, and in this case, it shows how ATP synthase is in the basically ATP synthase is in this picture. And something really cool is like ATP synthase goes from the thylakoid space. The hydrogen ion gradient goes from the thylakoid space because since the Calvin cycle occurs in the stroma, so it doesn't need to spend extra energy to transport ATP or NADPH. So basically, it's directly made in the stroma, so that it just goes directly to the Calvin cycle. No energy is needed for active transport. And so yeah, if they ask you a question about the acidity between the thylakoid space and the stroma for the chloroplast, then you're going to say that thylakoid space is more acidic because that's where more hydrogen ions are located. And so yeah, it goes to the Calvin cycle. So this is basically the light reactions. This is a summary of it. And then yeah, now after it makes ATP and NADPH, we can go to the Calvin cycle. And so the Calvin cycle occurs in the stroma, which is the fluid of the chloroplast. And it uses ATP and NADPH to convert carbon dioxide, in this case, to sugar, which is glyceraldehyde 3-phosphate, or G3P, and returns the ATP back to ADP, and then ADPH to N+, and then which can be reused in the light reactions. And so you have 3 phases of the Calvin cycle. First, have carbon fixation, in which you have you basically have carbon dioxide that enters, and Rubisco is basically your protein that will reduce carbon dioxide. So Rubisco is, fun fact, is like one of the most abundant proteins on Earth because of the fact that it's really crucially needed for the Calvin cycle. And so Rubisco basically reduces carbon dioxide, and what it does is it binds to a 5-carbon sugar. It's called RUBP, but you probably won't need to know specifics. So you just need to know, like, CO2, it gets reduced. And then basically it produces G3P. And then since it's a cycle, the RUBP, or basically like the sugars that you use initially, have to be regenerated. So yeah, you don't need to know specifics in this case, but just know that G3P is formed and that glucose isn't necessarily the final product of photosynthesis. Sorry, it's G3P, but that basically G3P can be used to make glucose. And so for one cycle, yeah, so basically, sorry, for 6 cycles, you can make 1 glucose molecule, and for 3 cycles of the Calvin cycle, you can make 1 molecule of G3P, and that's because G3P is a 3-carbon sugar, so 3 cycles for 3-carbon sugar, and then 6 cycles for glucose, and that's like an easy way to think of it. And so yeah, 9 molecules of ATP and 6 molecules of NADPH are used to make 1 molecule of G3P. And you don't need to know this, but like there's a thing called cyclic electron flow, and it's just a way to make, because you need 9 molecules of ATP compared to 6 molecules of NADPH. So extra ATP can be generated using cyclic electron flow. You definitely don't need to know that. It's just like it kind of helps with knowing why more ATP is needed and how it can be generated in plants. And yeah, so 6 cycles are used to make 1 molecule of glucose, which is 2G3P. Yeah, and so this is your diagram of the Calvin cycle. And basically, G3P is made every 3 carbons. 1 G3P is made. And so cycles will make 1 glucose. So that's basically it for units 1 through 3.